Good day, everyone. I am Gabriela Pio Barca, and today I will going to discuss concept of gene or gene concept. In this presentation, you're about to discover the history and evolution, definition, terms, classical view, modern view, genes as histron, recon, Newton, gene types, gene action, and gene in protein synthesis. But let us first sing together the DNA song in the theme of Row, Row, Row Your Boat. Make the nucleotides, sugar, phosphate, and a base bonded down one side. Adenine and thymine make a lovely pair. Cytosine without guanine will feel very bare. It is just an exercise for us to imagine or describe the important element involved in the concept. Further ado, let us now start a discussion with the history and evolution. This summary is organized with the year and the scientist behind the particular gene concept. Besides, I just want to reiterate Johansen, a pharmacist and geneticist. He is best known for coining the terms gene, phenotype, and genotype and first 1903 Pure experiment in genetics. Let us now move on to the definition. The gene is to genetics what the atom is to chemistry. We already know that the gene is the unit of genetic information that controls a specific aspect of the phenotype. The gene is the unit of genetic information that specifies the synthesis of one polypeptide. Also, genes are the fractions or part of DNA molecule which regarded as the genetic material. We already know the levels of organization from cell into DNA structure within the cell, the nucleus, and the nucleus contains the chromosome, and the thread-like chromatid when it stretches out, it has the nucleosome that protects the histones, and by that the sugar, phosphate, and base bonded by the sides as stated on the DNA song. By that ACTG that will determine the unique traits of an individual. Okay, let us now move on to more complex terms. First is recon. It is the smallest unit of DNA capable of undergoing crossing over and recombination. A recon consists of not more than two pairs of nucleotides, which also capable of being separated and exchanged with other chromosomes. Next is muton. It is the smallest unit of DNA which can undergo mutation. It can also change or mutated that produce a phenotypic trait. Newton also defined as the delimited to a single nucleotide. The third on our list is histone. It is the unit of function. It is the gene in real sense capable of synthesizing a polypeptide chain of an enzyme. The part of DNA specifying a single polypeptide chain is termed as histone. A histone can have 100 nucleotide pairs in length to 30,000 nucleotide pairs. It transmits characters from one generation to other as unit of transmission. And the last is complon. It is the unit of complementation from the word itself. So let us now proceed to the classical view. So many years or few decades ago, people believe or define gene as the unit of function. One gene specifies one character, recombination, and mutation. That paved the way to believe that genes are discrete particles inherited in Mendelian fashion that occupies a definite locus in the chromosome and responsible for expression of specific phenotypic character. When we say locus, it refers to position of gene changes or character changes. After several events, the definition and function of gene was introduced by Sutton, year 1902, and was elaborated by Morgan 1913 and followed by Bidge and Muller and others. Shift into three function or definition as genes can be transmitted from parent to offsprings. Genes may exist in several alternate forms called alleles, and genes are capable of combined together or can be replicated during a cell division. Moving on, let us go to the modern view. In the modern view, gene is defined as the unit of genetic information or the sequence of DNA that specifies one polypeptide that includes coding as well as non-coding regulatory sequences. Meanwhile, Benzer on year 1957 coined different terms for different nature of gene and genetic material in relation to the chromosome on the basis of genetic phenomena to which they involve. Bezer defined gene into three. Number one, genes as unit of transmission or cistron, genes as unit of recombination or recon, and third, gene as unit of mutation or muton. In this space, we can infer that we already defined these terms into the past slides. So after knowing the shift of defining gene from classical to modern view, let us now start to classify the gene types. On the basis of their behavior, the genes may be categorized into the following types: basic gene, little gene, multiple gene, cumulative gene, pleiotropic gene, modifying gene, and inhibitory gene. To further understand, basic genes is defined as the fundamental genes that bring about expression of particular character, while little genes bring about the death of their possessor, and multiple gene defined when two or more pairs of independent genes act together to produce a single phenotypic trait. So, as the cumulative gene refers to as some genes have additive effects on the action of other genes, and pleiotropic genes are the genes which produce changes in more than one character. And modifying gene is the gene which cannot produce a character by itself but interacts with other to produce a modified effect. And last, inhibitory gene, which is the gene which suppresses or inhibits the expression of another gene, is called inhibitory gene. We can easily identify or differentiate the gene types based on their behavior and function. Let us now proceed to the gene action, which plays a major role on the gene concept. The influence of gene resulting in the expression of a genetic character is called gene action. We can see here in the image from DNA, it is transcribed through mRNA and translated into proteins. The genes are generally associated with the production of enzymes which they synthesize from chemical substance available in the body cells through a process of autocatalysis. As a rule, one gene affects one enzyme. Their various actions of genes are expressed in their development of pigments, colors, hormones, size and form, production of proteins, antigen and antibody production, decisive effect on human disease like albinism, etc. Let us now discuss the last topic in the gene concept, which is the gene in protein synthesis. Some may undergo sudden change in expression called as mutant gene or mutation, or may be transferred to its homologous crossover or non-homologous counterpart or translocation. Can duplicate themselves very accurately are called replication. And the last is synthesizes a particular protein and determines the sequence of amino acid in the polypeptide chain is called the genetic code.